Good day, my dear students. I welcome you all to the second lecture in BCH 407 titled Chemistry, Classification and Nomenclature of Drugs. Today we shall look at reaction, categorization, and naming of drugs. In drug chemistry, we will be looking at two phenomena, namely theories of drug action and interaction of drugs with cellular components. Now, we have 5 theories of drugs, namely receptor occupancy theory, the rate occupancy, the rate theory, induced fit theory, molecular or macromolecular perturbation theory, and activation-aggregation theory. As usual, let's consider them one after the other for simplicity or better comprehension. This theory, that is, the receptor occupancy theory, is often explained by the law of mass action. It states that a drug effect depends on how many receptors it occupies. The more a drug is present, the more receptors are occupied until it gets saturated or until saturation occurs. This is a simple description of that theory. This picture describes the receptor occupancy theory with a few receptors and low or high concentration. Low effect of drug will occur as the receptor and drug concentration increases. So is the effect. This is because receptors are all saturated. You can see from my left here, you have fewer receptor and drug molecules. Now here, you see that the drug concentration has increased, and of course, the receptors have also increased. So you have more drug occupying the drug site compared to the third one, which is a very high concentration, where all the receptors of the drug are already occupied. So we can say that the rate and extent of chemical reaction are directly proportional to the concentration of the reacting substances. Now, the rate theory. This theory simply testifies that the speed of a drug receptor binding is central and not just occupancy. So this is saying that binding speed matters more than occupancy. Now from the left side, you have fast binding dynamics. Drug molecules rapidly associate with receptors and dissociate quickly. Association sends biological signals that produces effects. These produce quick onset but short-lived effects. Even with low overall rate receptor occupancy, a fast on-off rate, can still generate, a fast on-off rate, can still generate, pharmacological activity. On the right side, we can see that there is a slow binding dynamics. Drug binds slowly and dissociates slowly. This leads to gradual onset but prolonged action. So the more time it stays in the receptor, the longer the effect. Induced fit theory. This theory assumes that receptors alter their conformation to accommodate drug. Now you can see in the picture 1, 2, 3. You can see a drug molecule here. It's a drug molecule approaching, coming to the receptor. Then you see the drug initial binding or loose fit. The binding is weak, reversible, and partially fitted. Now you can see that the drug has induced conformational change where it is better fit and of course produce adequate response. You can see the drug here. Macromolecular perturbation theory. This theory explains that drug alters receptor structure in two ways. First, specific perturbation, where agonists twist receptor precisely to activate them. And secondly, non-specific perturbation, where agonists induce random changes that do not trigger responses. Again, this is a picture depicting what I just explained. From left, you can see agonist binding receptor in a specific manner, thereby twisting both the receptor and the drug molecules to trigger response. This is it. After binding, you can see how it has twisted to produce response. But this is not specific perturbation. You can see it. This is random distortion. Of course, no response. Now, let's look at the activation-aggregation theory. Again, this theory holds that receptors naturally toggle between active and inactive form or states. The picture you see here illustrates the two-state receptor model. It shows that receptors naturally and continuously switch or toggle between the active state, which produces signal or activation, and an inactive or rest state, which produces no signal. This is the active state, while this is the inactive state. This produces signal that generates response, and this does not. Again, that said, let's look at interaction of drugs with cellular components. Drugs exert their therapeutic and toxic effects by interacting with specific cellular components, principally receptors, enzymes, ion channels, and transport proteins. We shall again consider this in turns. But let me just expatiate a little on this. These interactions initiate biochemical and physiological changes that ultimately modify cellular functions and whole body responses. At the molecular level, drugs may activate or inhibit signal pathways, alter membrane permeability, modify enzyme activity, or influence gene expression, thereby causing one or more responses, that may be positive or negative. Understanding how drug-cell interaction affects the living system is fundamental to biochemical pharmacology, as it explains how drug produces desired effects, why side effects occur, and how variations in cellular targets contribute to differences in drug response among individuals. Like I said earlier, let's look at each of these in turns. Now, receptors are central to hormonal transmitters and immune signaling. You may ask, what are receptors? They are simply specialized proteins on cell membrane that binds drugs or ligands to initiate a cellular response. You can see from the picture here, this is a ligand, while this is a receptor. This is a cell membrane. Why the ligand fits into the receptor, you can see the shape of the second one. Of course, it will fit into it perfectly to initiate cellular response by sending biological signals. Now, how about enzymes? Enzymes catalyze biochemical reactions in cells. Many drugs work by inhibiting or enhancing or activating enzyme activity. For example, aspirin inhibits cyclooxygenase to reduce inflammation, while ACE inhibitors block angiotensin converting enzyme to lower blood pressure. Enzyme targeting is common in antimicrobial and metabolic therapies. Ion channels. These regulate the movement of ion across cell membranes, controlling electrical activity in nerves and muscles. Principally, the ions that are of interest here are sodium ion, potassium ion, calcium, and chloride ion. Transport proteins. Some drugs act by inhibiting uptake or efflux transporters, thereby increasing or decreasing concentrations of signaling molecules. For instance, selective serotonin reuptake inhibitors block serotonin transporters to enhance neurotransmissions. Transport proteins move substances across cell membranes, including nutrients, neurotransmitters, and drugs. Now, classification and nomenclature of drugs. Drugs are somehow classified on the basis of 1. Structure, and 2 action sites. Now, classifications and nomenclature of drugs on the basis of structure. Drugs are classified as structurally specific and non-structurally specific. For structurally specific drug, they act through precise interaction with target molecules. A minor modification of the drug structure can greatly alter its activity. You can see here the lock and key manner of binding. Now, if one of the components is structurally defective, binding cannot occur. For non-structurally specific, these drugs exert their effects not through specific three-dimensional binding, but through general physicochemical properties such as altering membrane fluidity or osmotic balance. Such drugs include halogen, cyclopropane, alcohol ethers, and other osmotic agents. When drugs bind to their biological targets, they rely on several main types of bond for such interaction to occur. The most common among these bonding interactions in drug targets include hydrogen bonding, electrostatic bond, hydrophobic interactions, van der Waals forces, and covalent bond. Now, for hydrogen bond, there are weak bonds that are essential for stabilizing counterparts or binding counterparts into the active site of target molecules. They help to stabilize the bond, help to stabilize the molecules, such as you have in the DNA. For electrostatic bonds, they are common and typically reversible, playing a critical role in ensuring drug bind with the necessary specificity and can be released when needed. For hydrophobic interaction, such drugs interactions are pivotal for binding of drugs to hydrophobic pockets within proteins, often enhancing drug stability and selectivity. Van der Waals forces. These are weak attractions that arises from temporary electrical charge difference. The forces occur between a drug and its target, and they can collectively provide substantial stabilization, especially when the drug fits comfortably into the binding site. For the covalent bond, this bond formation is rare due to their strength and irreversibility. Covalent drug binding is usually reserved for drugs intended to produce long-lasting effects, such as some anti-cancer therapies and certain enzyme inhibitors. They are hardly desired in therapeutics. Drug classification on the basis of action sites. On the basis of action sites, there are 11 drug types. We have blood-related drugs, under which we have anticoagulants, antiplatelet agents, fibrinolytic agents or drugs. The second is drug affecting or muscle drugs, which I refer to here as drug affecting muscles. We have the autonomic drugs, we have the local hormones, and we have drugs affecting striated muscles. You know that muscles are divided into two. We have the striated and the smooth. Number 3, we have the digestive system drugs. 4, we have the reproductive system drugs. 5, kidney drugs, dermatological drugs, and endocrine drugs. You can consult your reference text for more. In this lecture, we have highlighted the following: theories of drug action, drug target interaction, structural basis or classification of drug receptor bonding types, and classification by site of action. Once again, you can assess your own understanding of the taught topic by attempting to answer these relevant questions. In case you have 1 or 2 questions, first of all, consult your reference text, discuss with your fellow colleagues or students, and when you are still in doubt or lost, you can keep your questions till we see physically. This is the end of lecture 2. Thank you for listening, and thank you for your time. Goodbye.